let me tell you about a CPLC project between my lab and two theory labs in Illinois. Alex Mantiev working on computational biophysics and Jun Song on computational genomics. In my cell, I have about 2 meters of DNA. Then this be put into a tiny space of about 10 microns of nucleus. And that happens by first packaging the DNA into this structure called the nucleosome. And we have developed methods to look at the mechanical stability of this structure at defined locality at different positions along the nucleosome. We also have a method to measure the flexibility of the DNA of a very short length scale. And we have found that the DNA flexibility has a profound impact on the stability of the nucleosome and possibly on gene expression. So the goal here is to understand how physical properties of DNA may affect cells' behavior. Now we're trying to bridge different scales, going from single molecules on 100 base pair length scale all the way to the genome scale by linking to the modern genomic technologies. And I will also talk about that effort where we're trying to build the map of DNA flexibility at the genome scale. In the nucleosome, there are about 140 some base pairs of DNA wrapped around the protein core, making 1.7 turns. Nucleosome has to satisfy two conflicting requirements. First, it has to be stable to serve as a compaction of the genome, but it also has to be dynamic to allow gene expression and regulate gene expression. So my student, Tui, has been developing single molecule assays to measure nucleosome dynamics on the physiologically relevant levels of force. Main tool that we use is called single molecule FRED, for fluorescence resonance energy transfer. If you label a molecule with green and red dye molecules, when you excite the green, you get mainly green emission. But in certain conformations, they are close enough to each other. You get strong energy transfer, high FRED efficiency, and you get mainly red emission. So by looking at a single molecule as a function of time, then you can deduce relative distances and conformations in real time. Another powerful technique is called optical trap or optical tweezers. If you want chopsticks made of light, you can grab a particle and apply piconewton scale forces and measure the response and so on. So over the years, we have built an instrument combining the two into one instrument. So we can measure conformational changes of a single molecule using FRED, but as a function of applied tension. Coming back to the nucleosome, Michel Wang and Bustamante labs have shown that nucleosomes unravel under tension in multiple stages. At low forces, the outer turn of the DNA comes off the protein core. At higher forces, the inner turn unravels, and yet higher forces will allow the protein to dissociate from the DNA completely. Now, if you imagine labeling the DNA using green and red dye molecules, as shown here, initially they are close to each other, so you high FRED efficiency and mainly red emission. But upon outer turn unraveling, there will be a major reduction in FRED and maybe some additional change upon inner turn unraveling and so on. So to measure this, we chose to look at what's called the 601 sequence that positions the nucleosome very precisely at a single position. And then we put the green and red dye molecules on the DNA. One near the middle is called diode, and the other near the end. So we call this construct end-diode labeling, or ED1. In the 3D structure, the two dyes are very close to each other at the entry point of the nucleosome. So you should get high FRED. This is a cartoon version of that. But as soon as you unravel the DNA from the end of the nucleosome, you should see a Fred decrease. Okay? So this is proving the outer-turn unraveling, and we can tether it to a surface using a biotin, and then link the other end to a long lambda DNA, 50,000 base pairs in length, and then the end is then grabbed by optical trap through a bead. You can apply force and measure a Fred as a function of force. You can also move the dyes to different locations as shown, so that now they are at the bottom of the nucleosome, again very close to each other. We call this internal labeling. Here, you are not going to see a Fred change until you unravel dozens of DNA base pairs. So this will report on the internal DNA segment unraveling. This is actual data. So for the outer turn probes, ED1, inner turn probes, internal, we are measuring Fred as a function of force for many, many single nucleosomes. And the Fred value starts at high value because their dyes are close to each other. But when you get to about 3 piconewtons of force, Fred drops, giving you this outer turn unraveling. If you look at the inner turn probes, Fred persists at high values until now you go to 15 piconewton, and then it comes down. So this is actually very nice. It is fully consistent with what others have reported, that at low forces you see outer turn unraveling, at higher forces you see inner turn unraveling. So we are very happy that we can do these measurements, and we should have published the paper back then, 4 years ago. But we made a mistake of doing one more experiment. So we said, well, if this is true, if you move the dyes to the other entry point, then they should also unravel at low forces. So my poor student had to do that experiment. And then she found that Fred actually stayed high up to high forces, even higher than the inner turn. So that started the whole saga here that I will report on briefly. So anyway, if this is true, then instead of unraveling symmetrically from both sides, you have asymmetric unraveling or even directional unraveling, going from one side and all the way around the nucleosome. So we thought, maybe this is caused by the asymmetry in the configuration, because we have short DNA to a surface and long DNA to the bead. So we swapped this and then found exactly the same result. So it's not due to this surface artifact. And I said, okay, so if this side, ED1, is indeed weak and ED2 is strong, then if you move the dyes a little deeper from the weak side, that side should remain weak, but maybe slightly stronger. Now you get even a little deeper. And if you move from ED2 to the strong side a little deeper into the strong side, maybe that side will remain strong, but maybe slightly weaker. So again, my young student had to do those measurements. And so the data is shown here. This is a weak side, Fred as a function of force. Black curve shows that this side is unraveling at low forces. If you go a little deeper, 1.5, red curve shows also low force unraveling, but slightly higher. And if you go deeper from the strong side, 2.5, that is still strong. So now we are getting the feeling that this is a real effect, not an artifact of the configuration experiment. But eventually clinched the thing was when then she added even more data points going around the nucleosome. I've already shown you data for the weak side, middle, and then strong side. And then when she looked at even farther away from the middle, and here they all remain weak, and on the other side they all remain strong. So now we believe that this is real, and instead of going like this, nucleosome is doing something like this, okay? Okay, but then what is causing this asymmetry, and is there any functional relevance to it? We think that this is caused by the sequence difference between the two halves of the DNA sequence, and as you can see, nucleosome has a very tightly bent DNA surrounding the protein core. So the idea we had was the following. Well, if the sequence makes the the DNA more flexible, then the sharply bent conformation will be better tolerated even under tension. So that side will be stronger mechanically, and vice versa. So flexible will translate to strength. Okay? So, but how do you actually test this idea? Because you need to measure the flexibility of the DNA about the half of the nucleotide DNA, about 70, 80 base pairs in length. So we were lucky because my former student, Reza, had developed just such an assay. So here we have a DNA immobilized to a surface with two sticky ends that are complementary to each other. When you go from low sort to high sort that prefers the loop conformation, then you can measure how quickly looping reaction occurs. And then the idea is that we can use a looping rate as a proxy of flexibility. Faster looping corresponds to higher flexibility according to our measure, which is the operational definition of flexibility here. So what we did was to use that looping assay to examine the two halves of the DNA that we used, a strong half and a weak half mechanically. Strong half loop quickly in 27 minutes, and green is for the weak half. It took 190 minutes, about 3 hours. So according to this measure, the strong half is 7 times more flexible. Again, flexible means higher strength. So that was exactly what we predicted. But of course, this could be a coincidence. So we did a bit more measurements and in more detail. For example, you can ask the next level question. Okay, what segment of the DNA is defining the nuclear stability? The first thing we did was to swap the outer quarters of the DNA sequence, as shown here. You can swap this one with that one. And then we measured flexibility and nuclear stability, and we got exactly the same results. So basically, apparently, the outer quarters do not determine these properties. Instead, when we swap the inner quarters, now the outer quarters are the same as before, but the inner quarters was swapped. Then what used to be strong here became weak, and vice versa. What used to be more flexible in terms of flexibility became stiffer, and so on. So now we believe that it is the inner quarters of the DNA sequence that determines the flexibility of actually the stability of the nucleosome. So this paper, in the end got published early this year, so my student is finally happy. So we find that basically depending on the flexibility of the DNA sequence, one side can be more flexible than the other. And one naive way of thinking about this is that proteins such as transcription factors want to bind to the DNA, but when it's more flexible, it'll be having a more difficult time to bind. So it'll just go away. But on the less flexible side, it comes up more easily, and then it can stay bound for a long time. And same you can apply to DNA repair proteins or chromatin monomers and so on. Okay, flexibly strong. The next questions are, what sequence features determine flexibility? And if this is important for biologically, perhaps then you can look at the signature in the genome that reflects the DNA flexibility. Okay. So we are developing an assay that we call LoopC. The idea is that you start from genomic DNA sequences, either taken from the cells or chemically made, and then you can attach looping adapters to this library, and then we immobilize them to a surface. You have thousands of millions of different sequences, and then you go from low sort to high sort to allow them to form a loop. And then most flexible sequences will form a loop very quickly. And then if you add an exonuclease to digest the DNA, starting from free end, then the flexible sequences will be protected from digestion and therefore enriched in the final sample. And then you recover the DNA, sequence the DNA, map it to the library, thereby achieving, you can then deduce how flexible that sequence is by measuring the enrichment effect. Okay, so in the selection here, an enrichment factor is our measure of DNA flexibility. So here's an example of showing this flexibility index or enrichment factor for many, many different sequences, about 5,000 different sequences. And there's a distribution, and this is log scale here. And you can then pick one sequence here, and then do a single molecule experiment to show that indeed this guy is actually more flexible, moves faster than the other guy from the other side, and so on. So we think that the assays is working. So we then went on to look at the body yield sequences. So this is a famous paper by Jonas and Witten, mapping the position of the nucleosomes in yeast with a single base pair resolution. So we know based on their research where the nucleosomes sit in the genome. And there's a diet position. They published diet positions for more than 60,000 nucleosomes. And then what we did was we just picked the 10,000 best position nucleosomes according to their model, and then we wrote down 50 base pairs of sequence to the left of the diet, middle position of nucleosome, and then 50 base pairs to the right of the diet, and then we asked the company to make that DNA for 20,000 different sequences. That's the library, and then we perform loop-seq. Then you can then, based on the reads, you can rank order them, which one is more flexible than the other. So these are the top 13 most flexible sequences. You can stare at them. I did, and I couldn't find out what was going on. So I did. I then actually plotted just flexibility versus GC content, again for all these sequences, and found that most flexible ones have high GC content, and low GC content sequences have low flexibility. It looks like that higher GC content makes the DNA more flexible, which is, to me, a bit counterintuitive. And here is actually another way of representing the data, and where we can rank all the sequences based on flexibility, high flexibility and going down in flexibility. What you notice is that yellow and greens are showing up at the top of the chart, meaning that they are high GC content again make the DNA more flexible. And plus, we also see 10 base pair periodicity, which we are still struggling to understand. And we also found that if you look at this particular nucleosome plus one nucleosome, the first nucleosome that RNA polymerase will see during transcription, that nucleosome tends to present the weak side, stiffer side of the DNA instead of the strong side against the invading RNA polymerase in most of the cases, suggesting that maybe nature optimized the flexibility of plus one nucleosomes so that it's easier to remove the nucleosome from the chromatin at the very beginning. Okay, one more minute. So we also have done a fun experiment, what we call mismatch loop-seq. Now we want to see the effect of single base pair mismatch on DNA flexibility. So what we do is we pick 50 base pairs in the middle, and then we can insert a single base pair mismatch at each location, or possible combinations. If you have a C on the top strand, then you can have three different possibilities: C, 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 T, and CA. So then times 50, you have 150 different constructs in one library. And then you do loop-seq, and then you do flexibility for 150 constructs. You see these highly flexible ones, and they're all shown in the middle because in the middle they have the largest effect, of course, in our assay. And if you plot the data in the form of a heat map, so this is the original sequence, top and bottom strand, and this is the variance, how you make this single base pair mismatch library. Interestingly, we found that C-contained mismatches are the most flexible ones. Actually, the high-scoring ones in terms of flexibility, they always have a C as part of the mismatch. So here is one that you can see that these guys are all very flexible, and also in this direction. And the second interesting observation is that and the flexibility is actually not only dependent on the identity of the mismatch, but it's strongly dependent on the sequence context. And most flexible ones are actually found in the CPG context, C followed by G, which is actually where the DNA is methylated. And here's an example where we have CG in the original sequence, and here then we have the highest flexible mismatches, and same here. So that's the kind of thing you can also do using the loop-seq method. So let me just start here. Tui was my hero here, and strong collaboration with Alec Axmantiel and Jun Song's lab, and a number of other people have contributed. Funding sources, the size of the icon actually reflects the relative contributions. Thank you.
Okay, we have questions.
Thanks, that's very nice. One question I had, so it's really puzzling that GC should be more stable, but what I noticed is, it seems like, I guess I remember John Middleton giving a talk, and I guess also if you could go back to the plot, it seems like in general, your GC contents are actually pretty low, so what you call IGC is really 50-50 or...
That's correct. So this is not a random library. This is from Bodding's.
Right.
Average content is 38%. Yes.
So could it, just as a crazy hypothesis, could it be that 50-50 is perhaps, have you tried something that is very high GC, like 80% or so?
No, we have not tried that, but it's a good idea.
Okay, I'm sure that your terminology makes perfect sense, but I'm totally confused.
That's good.
So, as you just mentioned, GC obviously is stronger coupling, so you would expect, maybe flexibility means something else to you. But usually people believe that promoter sites are reaching AT so that the binding factors can bind there, and they're usually on the linkage, they're not inside the nucleus. So if you're looking for binding sites for regulation of DNA expression, actually it's more important to look at the linkage, and the stability of the nucleosome actually doesn't matter. Maybe it doesn't. I'm just confused. What is flexible versus non-flexible?
Okay, it gets to the philosophical regime.
Exactly. So that's why I would like to understand actually what do you mean by flexibility. You probably talk about persistent length. That's I assume this is actually what you mean.
Maybe not. Okay, so
maybe later on I can explain a little bit more.
So I think so basically when I say flexibility, it's actually code flexibility on code. It's actually our operational definition. How quickly does it loop in our assay, right? And it has a strong correlation with the nuclear stability that we measure. So we think that it's a meaningful definition. However, we don't understand the biophysics of looping reaction very well yet, so we need to do more experiments to find out. Now, I agree with you. When I first saw that GC with DNA is more flexible, I was a bit baffled by that. But when I talked to my colleagues doing computational genomics, he told me that it's been known about for 10 years that nucleosomes form better on GC-rich sequences. And the gaps, the linkers have AT-rich sequences. That makes sense because more flexible sequence can wrap around the protein code better, right?
No, I agree. I just was wondering because usually soft regions in the DNA are AT-rich, so that actually things can alter the DNA and bind things like that. And so
we think that's actually opposite. Yeah.
Trying to get what soft means.
Richard's answer was, he didn't explicitly state this, but the looping formation is not purely determined by LP alone, but it's how it's coupled with modes or additional modes that are not usually represented in theory. And so until that coupling is known better, you can't be quite sure of whether higher LP means lower, it only means lower probability of forming the loop, but the dynamics is determined by other modes, and that's what he's finding.
Yeah,
I'm just trying that we should separate the two issues of formation of the nucleosome with binding of factors that control it.
I agree. Yeah.
One quick question.
So, flexibility here, since you're going to use words, we don't know what it means. How much flexibility depends on mismatching and how much depends on the sequence you have. Basically, is the numbers, like I talk about GCAT, or it's really the mismatches is the main point.
Okay, so I forgot to mention that. So, people generally, think that mismatch will have a huge impact on flexibility because you have a bubble and hinge and so on. But it turns out that, you saw the peak. Basically, if you don't have a C as part of mismatch, it has no effect. Basically, as I said, there's no mismatch whatsoever. It has the same our definition of flexibility as the fully base pair DNA. So I was a little bit surprised by that.
So sequence is more important than actually. That's where the
I think in the end, the sequence has a bigger effect than mismatch. Yes.
Okay, let's thank TJ again. Great talk.